between the wisdom passed down by ancient healing traditions, anecdotal experience, and modern clinical trials, one thing is clear: mushrooms are medicinal powerhouses. And I finally found a brand, a product, a company that I feel really aligns with all of my research and understanding when it comes to the medicinal properties of mushrooms, and that is Alchemy Mushrooms. They grow their mushrooms in California on organic mushroom farms. They keep the whole mushroom in their supplements, and they actually blend mycelium and fruit body in their mushroom powders and capsules to give you the best of both worlds. You can learn more at Alchemy Mushrooms. That's ALCHEMI. alchemymushrooms.com. Use the discount code MushroomHour for 20% off your order. Alchemy with an I, mushrooms.com. Hi there. Welcome to Mushroom Hour. Today on Mushroom Hour, we're graced by the presence of Matt Powers, author, educator, citizen science, entrepreneur, and family guy who teaches people all over the world how to live more regeneratively. Personally driven by a deep desire to have the best food possible for his wife and cancer survivor, Adriana, and their two boys, Matt, a former public high school teacher with a master's degree in education, is creator of over a dozen online courses and author of over 20 books on permaculture and regenerative soil science. The Permaculture Student series and the Regenerative Soil Trilogy are just a couple examples. Matt is also the host of A Regenerative Future, a podcast and YouTube show where he interviews leaders in the regenerative space and shares his own work and insights. I'm excited today to learn all about the soil ecosystem, regenerative soil microscopy, and so much more. Matt, thank you for joining us on the Mushroom Hour.
Thank you so much for reaching out and having me on. I'm honored.
Well, seriously, the honor is all mine. Mutual friend William P. Brown was showcasing a bunch of your work, and I was like, who's this guy? What is all this work, this body of work, massive books and shows, and I need to learn more. So thank you for agreeing to come on. And to kick it off, what led you to be obsessed with soil science and dive into the rhizosphere? Tell us, the highlights, the arc, a little bit of that journey.
Well, to begin with, like you were saying in the intro, my wife kept getting cancer and I was trying to pay for the organic food, but I was living in my in-laws' parents' garage. So it was a situation, and I was a high school teacher, and every year I was in more debt. And so I was like, I can't pay for organic, but I must have organic. And then if you're doing the Gerson therapy, as we were for 2 years, it's like buckets full, bushels full of carrots. And so you end up having to pay a fortune or find a farmer who will sell you his funky looking carrots that he can't sell at market. That's what we did. But I realized I needed to grow my own food. And I also realized that my soil in my garden was better than the people we were buying from that was an organic farm. And I was like, what's happening here? The soils were decomposed granite. And so they look like little crystals, like sand. And they were 140 degrees in the sun. When we were in the Central Valley of California, right below Yosemite. And so it's bulletproof, like soil in the middle of summer, and it's just so hot. So people thought you couldn't grow there. And so everywhere and also, California has got some places with good soil. Like Oregon's got Willamette Valley. There's places, but most of it oxidized and alkaline and terrible soil. And so every place I've gone, I've had to move to and restart over. I've always had to focus on soil, and it was kind of hammered into me. So I was focused on the seeds, and I did the seeds, and then I was focused on design, and I did a design. I've gone all over the map. The way I learn, the highest level of learning is always teaching, and so if I want to learn something, I learn it all the way. And I have to have a fluency in it. And the only way to fully develop a fluency is to actually, for me, to design a curriculum that literally teaches people at a comfortable reading level how to understand and then implement those things. And so that's what I do. And so I've gone through and I've translated for high school kids, for elementary school kids, for middle school kids. My stuff's been translated into 6 different languages, the permaculture stuff. And then what happened was I went for 2 years on this deep update of permaculture designs, like the designer's manual by Bill Mollison. It was made in '89. So of course things have changed, right? Can't even find most of the other references for like anywhere less than a fortune, and they're all out of date, and out of print. And so I was like working on finding all that information. The biggest chapter in that book was soil. And when I asked people this last time before this one, what I should do next, because I'm still a teacher, right? So I'm like, what do the students want to learn? And I'll learn it and I'll translate it. Let's go. And so they let me know they wanted me to do soil. And so I was like, I thought we were going to do gardening, permaculture gardening, and I was going to like take my permaculture gardening course, which is a course I did. It's one of my most popular courses. It's very open and easy, but they wanted the soil. And so I delved into the soil, and then it was like a can of worms that exploded into my face. And all this uncertainty and all this like doubt happened. I encountered myself from high school again being like, you didn't do well in high school chemistry, Matt. And then I was like, wait, because that one day when she made fun of your stuttering. And then it was like,
The whole
I
journey.
like went back in time and like face old demons. But like I, in that process, created a holistic framework for understanding soil that had never been done before. So I took biology and chemistry and put them together because obviously the biology is doing biochemistry. Otherwise,
Right.
the plants wouldn't get any nutrition. Right? So I've got drawn into this for my curiosity, from its actual linchpin too, I would say. There's a gravity, like with fungi, you see it all the time. There's a gravity, a causal gravity, right, that leads us back to fungi. And I would say the same thing is here with soil. There's a causal relationship to health, to the environment, to the way that ecosystems manifest, to the way that microbes horizontally gene transfer and manifest themselves as expressions of the environment. Like there's so many layers to this, but it all revolves around these key leverage points. And from our anthropomorphic perspective, we've given them labels like fungi, and then there's like these things that are like somewhere in between, like oomocytes and actinobacteria and actinomycetes, right? It's because they're borrowing genes from each other, horizontal gene transfer. So I got drawn in, and my teacher brain loves to take things that are complicated and make them visual and understandable down to pragmatism so that we can actually do something about it rather than it just being nice.
Well, and I think a lot of people who have become interested in fungi realize quickly how important they are in the soil web, and the amount of people I've talked to who eventually tumble down into the soil food web because of wanting to study fungal ecology and lifestyles. And yes, I Kiss the Ground kind of popularized it for the masses, I mean, including myself, and just how keystone the soil is, and how, I mean, it's a beacon of hope, right? When so many problems or so many externalities can be traced back to soil. Okay, well, now we have a starting block. Now we have a starting point. And I guess then for you, kind of what is that framework and some of those levers that are determinants in soil health? I mean, we've referenced some of the organisms, and I guess you can go as deep as you want, or as, 10,000 foot view, but some of that framework that dictates, okay, what's in the soil? How do we know if soil's healthy or not? Things of that nature.
Yeah, for so long, the tests and the research has been done on crap soil. And so the results are pretty beguiling. They're not going to give us information that's useful. And so it's been a process of getting people to recognize what good soil is. And that's why I'm working on this database so that people, because everyone does these tests in black boxes. So they do this test. The extension center says to add this much nitrogen, this much this and that, and no one compares their tests or anything. And the hallmarks for health are never really discussed. And one of those hallmarks of health that very recently, but not in the mycology community, if you're connected with the right people, until very recently wasn't understood, was endophytes. So these are bacteria and fungi that live inside plants, but in the sterilized world of GMOs and synthetic chemistry, because they're zapping the soil with those salts as they disassociate, and they've created that flood of ions that are just purified, and so they want to bond with everything. So they just tear the organic matter apart, and they rip through the membranes of all the microbes. That's what purified nitrogen does, just like purified charcoal does in your forest soils. When they say like char can hurt the soils, like they're talking about doing that. That's why biochar must be inoculated first for the purpose of what you're doing. So they've been removing fungi with synthetic nitrogen since the 40s. And tillage also kills fungi, no doubt, but yeasts are everywhere, right?
Yeah.
But when you use antifungals, they aren't. So the structure of the soil is primarily fungal, either dead fungi or living fungi or the DNA. I'm finding in my DNA testing that there's a large amount of DNA. And I mean, like William Padilla Brown, right? He's the one who spent an entire day with me breaking DNA. It took a day, and then I had to do another day of work to break the DNA. And theoretically, DNA can last millions of years. So in other words, the soil is largely comprised of fragments of DNA, and the microbes are literally eating that and absorbing that and using that when it's useful, and then sourcing that from the environment. So when we say that microbes are an expression of the environment, that's how.
Literally, they're getting bio upgrades. Yeah.
And so do we, because we absorb 60% of what our touches our skin goes in, and we inhale, right? It goes in too. And so all of these modalities are overlapping. So one of the other hallmarks of really good soil is going to be fungi. And there's a generalized perspective on this, and then there's an emergent, more specific, more dialed in perspective that's emerging. And we're trying to figure out new test methods, because fungi is difficult to crack DNA. Actually resists it very, very well. So much so that even experts are saying that we might have to continue PCRing it, which is frustrating because that throws all of our numbers off. And then the reliability and the amount of error, right, in our results goes up. But fungi in general is pretty easy to see. It's huge. And then the yeasts are also pretty darn huge. I mean, in comparison to everyone. And so we can see these things very easily. We can document and take pictures of fungi very easily. We need to spread, because all right, so this is the thing. Like, people are using wood in their compost and doing wood saprophytes, but only 17% of wood saprophytes have been known to be endophytes. We should be selecting for those endophytes so that we're breaking down our wood smartly with this process of, it's like, oh, no, this is not only good for removing this wood, but this goes right into the plants. And so I would say that fungi is one of the hallmarks. Organic matter is twin to that. So it could just be that you're doing at home, just to get a general idea of like how you're doing. You can be doing a jar soil test and seeing how much organic matter. You can be doing a pH test, because above pH 7, things are going to be bacterial.
Right.
At neutral and below, things are going to be more fungal. And it's just a product of moisture, because pH is power of hydrogen, right? And then the microbes and the plants are performing hydrolysis, and so they're liberating those protons from water into all the surrounding receiving areas, whether it be biochar or humic substances, or another microbe, right? Or a plant root. And so life and organic matter are the hallmarks, the generalized hallmarks for these things. And then the next part is it's fungi, because some people have like life and it's just bacteria. And they've got that hot compost, it's high in nitrates, which means that it makes for big watery cells, because you need 4 times the molecules in water to process each molecule of nitrate. So it's an huge increase in water. That's why they put on the water weight. That's why they grow so big. And if it's more fungal, if it's more acidic, then it's more ammonium. And that's going to be more perennial. It's more stable, and that's going to be fruiting and reproductive time period on a growth scale. Because nitrates are okay if they're at a certain level and at the beginning of plant growth. Really, it's life and organic matter, but there's these new layers that are so critically important.
Well, even just this idea of packets of DNA being available in kind of the growth matrices as being important. That's something I had never really heard about or picked up on. Selectively choosing endophytes. I think a lot of people, we hear about endophytes, one of the big question marks is, what is their role? Are they just riding in a much safer habitat? Are they imbuing tolerance to the plants? Are they?
So the only
it's
way
really
that an
interesting.
endophyte exists inside of a plant is it has to fix nitrogen, because all the microbes inside of a plant are bombarded with superoxide. So they have to be able to fix nitrogen and then give it to the plant in exchange to stay there. So they form these little like halos around themselves. Really, it's 3D. So it's like a force field around them as they're being bombarded. And you can actually take, one of the steps is peroxide. One of the steps is blue tetraosolium. And I forgot the other stains. But you basically can stain a root, and you can watch every stage of this process. James F. White, many of you in the mycology community know this amazing man. He's the one who, in America, described rhizophagy. It was done once in Australia. He verified it, replication, right? He's the one who's been spreading the word on it. And he shares every single stain in his papers. His papers is James F. White. Rhizophagy is the word to look up. And he maps it all. And he tells you exactly how to, and he gives credit to each of the students that does each work, and their pictures are in there. This is the classic, the kind of teacher that like the movies are made of, you know what I mean?
Yeah, that's
And
right.
so yeah, we all can do that. We all can witness that this process happens. And so I've actually seen them coming out of root hairs, the bacteria being, because that's the end of the rhizophagy cycle, is they actually regrow their outer membranes and then get pushed out of the root hair. And root hair growth only happens when roots are eating microbes and there's endophytes giving that nitrogen. So it's really, really absolutely amazing what we can learn at this juncture and what we've already figured out. So endophytes, what they're doing is they're doing 2 things. They're fixing nitrogen, but when they're in there, because they triggered that plant to go attack, the plant goes on immunological guard and alert permanently for the rest of its lifespan. So you've triggered an immunological response. This is what like natural farming is doing with like their OHN. They're shocking the plant. The plant's going, ah. And vinegar can do the same thing. Acetic acid can do the same thing. Shocking the plant, and the plant goes on guard. The microbes do that too. But the microbes are also doing secondary things. They're sopping up excess sugars. Like for instance, they're finding cerveza yeast in everything. So alcohol was going to happen no matter what. It probably happened over and over and over and over again until we like figured it out. And we're like, I know what's going on. It probably took a while. But like cerveza yeast is in everything. And so it eats sugar. So if a plant has excess sugar because it's a sunny day, they help.
Propagates cerveza yeast.
And within us, we have a digestion that helps us have an equilibrium in our body too, right? We have an immune system that helps us have an equilibrium. And even in our cells, mitochondria was an invader was once an endophyte. So this is all part of the pattern. This is all stuff we all kind of know. And it's just we're getting introduced to another way that it's being manifested in our world. So yeah, no, I'm really, really excited about all this because they're also releasing biostimulants, and that become either they are the plant secondary metabolites that are the medicinal compounds in plants. That's one argument, or they're stimulating the plant to create those at the highest levels of health. We know that that's what they do, and they're all there at the same time. There are some examples of like certain microbes creating beta carotene and doing those kinds of things. But I don't know if I can say that all of them are doing all of that, right? Some of them might just be hanging out. Some of them might just be sopping up sugars or turning nitrogen into amino acids.
That's one of the things I love to say to blow people's mind is, oh, all of these plants that we think have certain properties or compounds, it might be coming from the fungi inside them. But I appreciate you deeping that even further to say that might not always be the case, or it might be stimulating the production of that within the plant. And so, in this model, I didn't expect this to turn into a conversation about endophytes, but that's kind of incredible. And I'm not surprised. Endophytes are really, in many ways, the frontier, it sounds like, of soil science, but also of fungi mycology. Some of the most seasoned mycologists I talk with just know that endophytes are going to be this massive focus. And right now, dark endophytes, there's all kinds of things. I've had Rue Vandergrift on the show, talked about looking at endophytes in grasses and things that we don't even have names for and can't readily identify. So it sounds like more of those in a soil ecosystem is going to allow plants to have allies that will help them in uptaking nitrogen and fixing nitrogen. They also may be able to do some kind of horizontal gene transfer and enlist that endophyte as part of their biology, basically, which I guess they are, even if they're just in there fixing nitrogen. They're able to enlist these endophytes as helpers as part of their biology. Is it that simple, though, of just adding more endophytes? The only reason I bring that up is I know there was an obsession when mycorrhizal fungi and their potentials were first realized. And people who, like myself, like everyone, gets excited about applied mycology. We're like, inoculate everything, use commercial mycorrhizal inoculum, spread it everywhere. And then I had people on who were actual applied mycologists, not just people psyched about it, who said, well, we don't really want to do that because we don't want to necessarily displace native players. So how do you see us best be able to harness endophytes and kind of, is it cultivating them in environments? What does that look like? Kind of use these endophytes to their highest potential, but not like abuse or overuse.
Did you talk to him about dark septate endophytes?
That's what I'm thinking of, not dark endophytes, dark septate. That is correct.
Yeah. So have you seen any of my work where I show how plant cells really look like glass? Like, of course, this makes sense because how else would sun get in, right?
I
But
haven't seen
that
that.
means like the sun passes through in the right wavelengths, right through and hits that microbe. And if you are dark septate endophyte, that means you've got that super black carbon inside you, meaning the sun is hitting that and heating that up. What does that do energetically?
Right. What are the implications of that?
Yeah, it's very, very fascinating because we were talking about, all right, so to backtrack, your question was,
How do we kind of have an applied use then of endophytes? Because I love that idea that they are a key to enhancing soil ecosystems. But how do we do it in a way where, we don't necessarily want to introduce, or maybe it's okay to introduce some like commercial strain?
diagnosing what's there, and so you understand who's there. And then number two, I've actually tested this out. So I've got a few different examples. First, like let's take EM. So everyone's like effective microbes. They are lactic acid bacteria, lactobacillus. They are streptomyces. They are um, purple non-sulfur bacteria, Rhodocytomonas palustris, and cerevisiae yeast. And so everyone's like really like wanting, you know what I mean, wanting to know if it's okay to have those things. I find them in all the best compost already.
Mm.
And then you can buy it as an actual body, like a probiotic for the body. So it's edible. And it helps and they're all the ones that are listed are endophytes. So they go in us, they go in our digestion, and they guide all of these things, or they're hallmarks of good digestion, good decomposition, and good plant health. That kind of like either or chicken and an egg thing is super interesting. Because Dr. Brzezinski, the urine doctor, the cancer doctor that was curing people with what was in healthy people's blood,
Yeah.
it's the same kind of thing. If we just bring back the biology, it brings back the health. I think the biology was the immune system of the plants. It's their feedback loop with the loop with the soil. And so without it, they actually, and I also think that there's like a paramagnetic tie-in too. I mean, like hearing that like microbes collect to an area and then that's where lightning strikes in a forest, that's pretty wild. I learned that this week.
That's a thing?
That's a thing. Quatamok View was telling me about it. And so it's like, we don't, I mean, you know that like purple non-sulfur bacteria eats electrons and there's other microbes that eat and share electrons, right? And that makes sense because oxidation is measured in millivolts. So you add oxygen to something, you dry something out, and you change its actual conductivity, which kind of makes a lot of sense.
So are these players then pretty ubiquitous across soils we look at? So maybe we don't have to be as concerned, like in my
Right.
mycorrhizal fungi example, we might have vastly different players in different forests, but in this example, we're finding the same endophytic players largely, no matter what soil you look at.
And not only that, there's 10 different major saprophytes that are in every single sample I find, even native soils, meaning that
Mm.
it's just a difference of a ratio between like rhizobia, streptomyces. E. coli is 50% of everything. And everyone thinks E. coli bad, bad. There's like what,
Yeah.
12 pathogenic E. coli, and there's millions of commensal endophytic E. coli. They're in all the plants. They're already in our digestion. We know that, right? Because everyone's like, oh no, careful, feces, E. coli. You know, but it's like, where did it come from, Johnny? Where did it come from? And it came from the actual plant. It was already in the plant to begin with. And same thing with like rhizobia. Rhizobia doesn't leave the decompositional event, even in the heat of the hottest compost. Rhizobia doesn't leave. So, in other words, rhizobia is traveling through all the ruminants. It's in their digestion already. It's their probiotic. And so, if you've got good compost and you had ruminant and they've been eating like anything leguminous, they're going to have rhizobia in there, and you can use that as an inoculant on your beans and your peas and all that.
Oh, okay.
And so the mesorhizobia, bradyrhizobia, Burkholderia, even like all the nitrogen fixers, like they're all in there already because they're primary decomposers. But no one comes close to E. coli. E. coli is it reigns supreme. And I was reading all these things on it, and then I watched this really incredible, like summative moment video for me of this guy who is a genetic engineer. And I'm not like, I'm GMO free. I'm like, that's how I am, right? I'm the hand tools guy. But understanding their perspective really allows me to understand what they're doing. And a lot of those guys they're poaching characteristics from microbes that no one's paying attention to. And so I can identify like the wild microbe that they poached from. So I read these things, I read everything. And so this guy was like, E. coli is a universal chassis. And he describes the number of genetic slots that are changeable. And so E. coli is constantly going with its genes and taking from the environment what it needs for that moment. And so it's constantly changing its species. And so I like realized because of that, we can't really put these hard and fast rules on biology, right? On like this IMO, and also me finding purple blood silver bacteria in compost in various areas suggests that it might be in the air. And when we aerate things, it might be adding it. So I need to DNA test the air as well. But the EM I tested that was 20 years of extension, meaning that they kept brewing the EM over and over and over again instead of getting a new batch. He used living native waters wherever he lived. All of that's different.
Mm.
And it's like lactobacilla, and then it's got like a totally different end name. So the original lactobacillus is all gone. There's like a teeny bit of lactobacillus casei, which is the milk one, and then everything else is like wild native. That's the same family, right? They're bacilli family first, and then they're lactobacilli, but they're wild variants of the same exact. And I actually did a side by side DNA sequence of this to show it. And the original was half lab, half half E. coli, and the second one is like all E. coli and like a teeny bit of these wild lactobacilli.
And is that an example then of the environment? I mean, that lactobacillus base is exposed to that environment, horizontal gene transfer happens, boom, this is what we get there now, which then is, like you said, kind of bring it back. And this always happens in community ecology. It like feeds in on itself. That new native lactobacilli is then playing its role in any subsequent horizontal gene transfer to affect surrounding organisms in that same way.
Yeah, and then there's weirdness though, and you probably have heard of stuff like this. I mean, the way genes work are pretty weird. So I heard someone say that like the lens on your eye has the same genes as algae in the ocean. This was I forget his first name, but Hannon, the plant breeder. I thought that was kind of weird. But William was PCRing something and chopping something up, and it read as bacteria, and it was only fungi as pure.
Hmm.
Yeah, and then the whole thing with like human DNA being in everything. And it's like, but is that the DNA that transfers through everyone and we all share?
I was going to say, is that like a universal biological store of data?
Exactly. And so it's like we've categorized these things, but it's not none of these things have boundaries that are non-porous. Everything is constantly transferring between all of these different categories that we've created. So we need to do a lot more testing, and we need to do it all in a way that's transparent, because right now people with powerful AIs are going through every single database and stripping them. And so they're going through and going, and they're gathering up all the information they can, and they're training these AIs to be able to read all the information that's out there. Like I did this not for an AI, just for my brain with nature. You can go in and get a nature subscription, and then go and type in soil, and then bacteria, and then fungi, and like go through all the different keyword permutations and find all those papers and just download them all. But the AI is working. These AIs are working their way through all the databases, and they're collecting information, and it's all behind walls. It's all not accessible to us, and the universities are not going to be included. The university is going to be poached.
And what is the output of that? I mean, they're searching all these papers to be able to have a corner on some market of understanding biology
Well,
better,
two
kind of.
things. Meta studies are the new main way that people do things now, because they're like,
Right.
okay, I won't have to do anything. We'll just do comparative studies. I did this in college. It's like they don't have to do any really hard work. And I get it too. There's a huge pool of data now. but then the data is so questionable to do it at that superficial level, and trusting that like the 1,000 has an accuracy that you can like step on and put your full weight on. You really have to go to the individual studies and then see how they did it, how they measured it, and then as many of the variables as they'll share. And so transparency has to be, all right, the only way that we're going to figure certain things out is if we do it all in front of each other and we show each other our work, and then we create a way that we can hold things up in alignment. So like just for like fluency's sake too, like let's say you've got a nematode and you're like, or a fungi strand, and you're like, look at this spore here, look at this spore pattern here. Is this the dangerous aspergillus, right? And so you can't even remember the aspergillus, variety. It's fumigatum. And so I had to look it up. What you would do is type in aspergillus pathogenic into the database, and then it comes up with 200 pictures. And you're looking through, and the pictures are rated on interpretation separately from quality of the image. So you can be like, oh, I don't want quality, I want accuracy. Click. And then it toggles for that. And you're like, all right. And then you just go to your image, go to that. You go to your image, you go to that. That's how you actually develop fluency. That's how we always develop fluency. Because it's like, at first, every dog was dog to all of us as babies. And then we were like, enough dogs passed by and we're like, that dog's crazy. Like, that's a pug, dear. Like, that dog's crazy. I love pugs. And then it's the pug dog, right? And that's what needs to happen is we need to take ourselves, and then also I want to, once we get to a certain level of funding, train an AI alongside us. Like, because we'll do the interpretations first, we'll get all the information, but then have us teach this AI, and then like have it like color coordinate things with spectrometry. And then like look at the times of day, because I think that there's behavioral differences in the times of day, the moon cycles, all of these things. Because if the tide is affected, wouldn't the things that are like basically made of water
Made of
be
water?
affected? Right? Right. And so, but that's so subtle. There's so many subtle things that these folks are mapping.
And I guess that's what computers are made for, is tracking many more variables than a human could in any reasonable time scale. And so I guess that gets into the project. I mean, your project is exciting in that you are creating, it sounds like, this open source citizen database. And I guess just hearing from, my kind of limited understanding of what you're laying down here, just hearing about these players in the soil ecosystem, what kind of endophytic players we're hearing about that are the common denominator in healthy soil that we're looking for, that we're trying to cultivate. I guess then why, and this is a very kind of nescient question, but why is it important then to have a database of many different areas if kind of the applied solution is drawing from the same set of 10 organisms? I guess, is it kind of a how much and where to apply these things, or kind of what is that reasoning behind developing this database to include samples from all over to see kind of those differences and everything?
There's a variety of reasons, but you'll see different ratios. Like that's what I suspect is the ratios actually deal with the plant type and deal with the climate and perhaps even the soil types. And so this is the thing. The reason soil science is kind of clunky is because they're all siloed out. It's like the chemistry department doesn't talk to the biology department, and they never have. And so when I combine those things, it opened up like a whole mess of questions. And so no one is connecting the plant health to the health of the soil in an open way. The only way to really do that and make it not say we're cherry picking, because I could do it as an individual, but how strong is that? That's a floor that not even I want to jump harder than that, right? Because it's just one person. But if it's a community of us proving that high organic matter, mineral coherence, so we're still going to do those standard mineral tests too, but we need it in context because it's only good for reliable for the most part. It's generalized on the others, but it's reliable for only like 5 indicators. And so mineral coherence, the biology, the organic matter levels, the CEC, the salinity levels, the pH, of course, the redox.
Yeah.
But even wilder things like the paramagnetism, the time of day that it was acquired. And if it's compost, it's like what substrate did you compost? Because that's one of the number one things they don't track is like what substrates go into the compost that lead to the biology and the nutrients you have at the end. And so but we haven't even gotten to the plant yet. So we're still on the soil, and those things have never been aligned. And the Haney test and all those, but like, let's bring them all in and put them together and showcase, because the only way you know the accuracy of a test is tested against other tests. And then the bricks of plants, which is the generalized plant sugar levels, but also the bionutrient meter, which is a spectrometer. And they're calibrating much like I'm creating this database, like they're doing a database that's relative. So the more people that participate, and the same thing will be with my database with the relative levels of global organic matter level percentage, right? And so there's going to be, we're going to track the global organic matter percentage bioregionally and globally, because organic matter levels are, A, that's the carbon, but B, that's the battery. That's where all the energy gets held. All the protons that come off through photosynthesis and hydrolysis inside the plant, they combine with the sugars, the carbonaceous sugars, and make humic acids. And so it's like, this is where the energy comes in, and it's how it's held, and it's actually how we expand the capacity of the soils. Without organic matter, you can't hold excess nutrients in a buffered capacity. And not only that, you can't take on excess electrons and protons, because otherwise your redox will go too low, and then you'll have soils that are too acidic, and then you have plants get diseases. So, yeah.
I was going to say, so there's 2 parts of this. So there's kind of establishing causal relationships between these soil dynamics and properties and the health of the plant. But then there's also this other objective of creating a map of those conditions over regions. I mean, once you get enough observations for a region that you can stand on, then you can say, okay, this region shares a commonality in that its soil has, a low carrying capacity in terms of adding more nutrients, things of that nature.
But on even further, think about how compost companies and farms are going to react to this. They're like, wait, this exposes the health of our soil and allows us to track it over time and compare it to itself for free. Okay. But then it shows their customers that, hey, my carrots have the highest bricks in the entire county, and my soil has the highest organic matter. And meanwhile, on our chart, they're number 1 for that bioregion. Highest organic matter levels, highest bricks level. And we're going to be rotating out these things because there's no one indicator of what is best, which is great, because there can be lots of winners, but it's lots of winners doing the right thing. And so this system is the first time in my life that I've created this very clean kind of almost magnetism in a system that will force like the people who are growing our foods to be more transparent, to do the right thing, and then be honored for it. And then at the same time, honor everyone for doing the right thing and expose the discernment of good, better, best, so that we can progress faster and faster and faster towards us all having more and more regenerative soils that get better every single year. I think this is actually a model that we're going to see spread. And I think we'll see it in mycology. Maybe William will do it, but then maybe we'll do it together as part of this and connect them, because so much of these things just connect in the soil, especially with like ectomycorrhizae, because they're literally mushrooms for the most part.
And physically, a lot of times they connect. Yeah.
Yeah. So this creates an imperative. They're drawn to do the right thing. It's like a magnet. And so they're like, oh, I have to compete, and it's going to force me to expose my thing, but then I'm going to get better. And they're like, it's like the best pressure I could ever think of. And then for the people already doing it, they're like, oh, this is my moment. Right. I can just go on here And that's why it has to be free, because I want everyone to feel, and anonymous if they want to be. So they can drop a pin for their site within 5 to maybe 10 miles, I don't know, maybe 5 miles of where you live or where the site is. So you can be anonymous, but we track where these soil types are, because I want people to take pictures of their soil, because like with spectrometry for fruit and leaves currently, I think it's going to shift over to spectrometry for our soil and compost, and it will be able to read. And I've got a polarizer lens right here, and I'm going to be getting later this month, I'm going to be getting a reference slide kit that will allow me to map out the actual frequencies, well, degrees on the polarizer that, and it is a frequency, that allows for the certain bit of light to pass through, that allows for each element to shine. So what I'm going to do for my students, because they're like $350 kits,
Yeah.
but I'm taking 4K pictures of them. So this is unbelievable quality, and that will be their reference. So they don't need to buy the slides. They will have, and then everyone else who's got those physical slides too will go, oh, I have the same kit. I'm going to do the same thing. And then they add theirs. And of course, every slide's different. Every prepared slide's different, right? And so we're going to have all these reference slides for minerals. So people with polarizers can see the minerals in their soil and then the compost. We're going to bust down the doors that are like on a lot of these things and connect them. And once there's freely flowing information between these things, and that's why I said like we'd have an AI as a member alongside of us, because we're all going to be allowed to reinterpret the entries. Like members will be allowed to reinterpret the entries and it's like, actually, I think it's like this. And I want to encourage kindness, but also to help each other, because only in the discussion do we get better. I had this guy tell me, like, because I don't like using eyepieces. I get that. I get the headaches. A lot of people get headaches from eyepieces on microscopes. And so I do like a monitor and everything. And this old timey guy was like, you don't need to have a screen and to capture things to like work with people. They can just hop on the lens with you. And I'm like, no, we kind of need like the whole community involved. There's this a good friend of mine and student in Germany. He was working in his lab on a sample, and a smart farmer had sent him and another lab the same sample, and they came back with different answers, different recommendations entirely. And then they did it face to face on Zoom. Same thing happened to them. And so it's like trained, certified lab owners. They're a commercial company, both of them, and they're still hitting that. And for me, that was like, that's exactly what I'm seeing, like on my end. And I think that there's a lot of silence and a lot of people being like, feeling really alone, because they're like, I guess I'm just not seeing it. And then they feel bad about themselves. But the reality is that like only 0.01% of nematodes have been identified. And that was from a study, like I think in 2021 or 2020, very, very recently, could have even been this year. But we're just scratching the surface. We're just starting out, just like how mycology has exploded in the past 10 years. Soil science, because we've been not doing it wrong, we just hadn't holistically done anything with it, it feels like. And then the microscope, it's like none of them had photography friends, or the ones that did went and got the $50,000 microscopes. It's like no one with no money and a photography friend got into this or something. Because for me, like this microscope's absolutely incredible, but I have had to figure out how to turn this microscope, which is $250 new, into that microscope for my students. And so they could see the fungi, because fungi glows in the epifluorescence.
Well, and this leads to so many questions. I mean, first of all, the implications for biodiversity studies, the implication for increasing everyone's fluency about, I mean, that's all baked into this. Apart from the incentives to have producers, I mean, it really helps the community science community to become more familiarized with these concepts. But you just hit on the big thing I had a question about, which is kind of database coherence. How do you have to get people prepared to use this database and to submit data that is of the appropriate type and with the appropriate kind of level of, I don't want to say discernment, but the appropriate level of processing and everything of that nature. And then I was curious what tools or what information was included, because myself, I haven't done soil tests before. And,
Yeah.
we just moved to a new house about a year ago. That was the first thing. We're like, all right, we're going to test this soil. We're going to see what's in here. And the options we found were kind of like, there's a local permaculture garden place near us in Sonoma. And it was like, oh, go bring us your soil. We'll go get you what you need to tell you what's in it. But it's, I guess, as part of this database and these holistic data about what's in the soil, what's in the plants, what does that involve kind of reagent wise and process wise? And then how are you helping people to be able to get to that level to where they can make meaningful contributions?
Absolutely. Well, that's, it's funny. I started at the end. I wrote a book that was going to update a lot of what people are doing with soil science and microscopy. I wrote most of the book, and in the process of writing Regenerative Soil Microscopy, which is actually what started this Kickstarter, I usually do books and courses, and that's what I've always done. But I realized I was developing a soup, an ocean of information. I mean, even today, I went through an hour and 15 minutes of dark field microscopy video. And when the video comes out, it's going to be like 7 minutes long.
Yeah.
And so people don't understand what it does. And so it's like, it's so much information. And I was live with a bunch of soil microscopy enthusiasts. And I was like, has anyone here come up with a good way of cataloging all their videos and images? And everyone went, there was like this like murmur. And I was like, this is what I'm talking about. You don't have a way of reviewing it. You don't have a way of cataloging it. You don't have a way of seeing things over time. And so I wanted to create that, not just for myself and my students, but for everyone, because this is the only way that we can like prove that these things work. Because if everyone's like, this is the biology, and then I do the biology, and then it's great. And it's like, what happened to the minerals? Where are your minerals at? You know what I mean? And then like, what's the actual biology? Like who actually was in there? Because there's people with the perfect fungal to bacterial ratios, and they've got certified laboratories, and their tomato plants with highest levels of biology are doing terrible. There's published literature on this. And they're commercial farms who've been doing it for decades. And they try to do transition to the biological farming. So it's imperative that we drill down to another layer of rigor in how we communicate around these ideas. And the great thing about it is it's not that hard if you make certain things clear. And so that's what I started doing. I wrote this book to update all the protocols to make it super clear. And I went to the university science, biology labs, mycology, some of the people practicing soil science, and permaculture. And I realized that like all the protocols like had principles behind them. And so they're like experiments, each one, just like we would learn in science. And so I realized like I could create my own protocols and going through everyone else's. And I also can teach other people how to do it too. And that's actually the right thing. And then I realized, I was like, Matt, you opened up a can of worms. But I was like, but that's the only way to actually count your worms. And I was like, oh my gosh. And then I realized, I was like, how else are we going to measure the relation of pH across bioregions in relation to nematodes or different variations of protozoa, or the species switch, or like all these different things? They're connected, no doubt.
Yeah.
And so, but no one has ever been able to align things well. And so I wanted to create something that was incentivized so that people would be like, oh, I'm always putting in my soil data up here because it helps me keep track of it. I actually can go and look at last year and see how much better I'm doing. And they get that reward. They can look up anyone else's entry. They can look up anything like nitrogen, and then it's ranked by nitrogen. I want it to be so useful that, and this is one of the stretch goals. I haven't like officially revealed this, but I've got to figure out how much it's going to cost. But I want to make it automated so that the counting of bacteria on slides is easy, because that's like the bane currently of folks doing the soil like consulting. They've got to like count the bacteria, and then fungal biomass and bacterial biomass calculations. It's like that all can be automated, and then inoculation rates of roots, that all can be the calculation of it can be automated. And my whole thing is, I'm like, I want it to be like, if you're a consultant, these tools are undeniable. And so for like a pittance annually, you want this. And so you're part of this. And then you're putting your data on here to keep track of it. And then it's public. So then people can rate you, and then people see your skill. And then people are like, hey, you're really amazing at this. Will you come consult because you're a consultant, And then it empowers people. It highlights people. It allows people their businesses to grow for doing the right thing. So I've never created something that had that kind of exponential benefit. I'm really excited about what it means.
Well, and I'm really excited about the concept because it's something that I've not really heard anyone doing. Maybe this is, just me not being deep enough in these circles, but it sounds like microscopy is the most kind of rigorous way to look at what's in your soil. And that makes sense. You're getting a visual what's in there, not just, we sent it off to a lab, they ran it through a number of compounds, extracted, isolated, even though that's more DNA. But anyway, they've kind of done these chemical processes, got a readout, but it sounds like microscopy is then even more definitive. Like, no, here's what's in there. Now, organismally, I would imagine you would see those players, but it sounds like with staining, things like that, you're also seeing minerals, nutrients, things of that nature too.
Yeah, especially when you can have like epifluorescence. So I can flip on the epifluorescence and I can see evidence of fungal activity, and I can see fungi. I mean, fungi, they glow. And their activity leaves glowing crystals in their wake. And so you can like imagine, because it's the wavelength that reaches them in the soil 2 to 3 inches down, that for a microbe traveling around, if you're riding on the surface of the hyphae, it's a glowing green highway. And you're just riding along this growing green highway. And they're the things in three-dimensional. They're these three-dimensional highways that lead through all the soil and lead to all the plants. It's wild.
That is wild. Well, then I guess, this project to create this open source database of regenerative microscopy soil information. Tell us about the Kickstarter, how people can support, what the goals are, all that kind of thing.
The Regenerative Soil Microscopy Kickstarter is a book and a course, and it turned into this database as well. So it's 3 things in one. Never done this. I've written so many books. I love writing books. I'm a writer. I was an English teacher. But this book is so different because it's the understanding and the tools and the know-how, the lens to navigate an entirely new world. Because not only are we doing bright field, but we're doing dark field, we're doing epifluorescence, and this technique that I've called light field as one word, but there is a light space field, very specialized, expensive lens that someone came up with and patented. It's very hard to find, actually, and I found it. So I don't think I can call it that, but it's using natural light, and it actually changes the way it makes color correct images. Because if you think about like what we're doing when we're shining a light, blasting it from below through things, in order to get to shine through it, you have to turn that light up a lot. And those microbes actually never experience light at that level. It's always low. So I've been increasingly using other ways of visualizing so that we can actually see things at much more color correct and accurate, that is like with the microbes experience. So I've been pioneering these different soil test methods, different visualization methods, as I've been combining all these best practices from biology, from mycology. Mycology, they are the leaders in so many ways in microscopy, which is so amazing. That's why there's so much overlap. And so I wrote the book. I haven't written the conclusion and I haven't done all the images. I want to get a few more images. I think I may have actually gotten one of the images that I needed today. But this book and this course were this way that I was going to train everyone to be able to evaluate their soil, to be able to use microscopes in a new way. And then it inspired the database. And I think the database is rockets. I mean, there's never been anything like it that I know of. Someone commented that this is somewhat similar to how they map space. As a community, they submit sections, and so that's super cool.
As above, so below.
Yeah, it's a proof of concept, and it's a community-based resource that's free for everyone to take part in. The membership allows you to compare anything to anything, so you can create like really wild metrics, especially once we figure out how to get it to read the top 50 or top 100 DNA sequence results. So if we can run the DNA against the biology that we're seeing and against all the nutrients and everything, we can actually get a more comprehensive picture of what's going on than it's ever been documented by anyone. And it'll be ours, like a public library. So this will be a place that universities will start turning to and then feeding into. And then it will cause this like trophic cascade, or this funneling of energy into an exponential change, a sea change. And what's going to happen? I mean, I already know what's going to happen, but I'm so excited about it. We're going to have organic matter levels jump and become ubiquitously higher all over the world, which is going to mean that our soils are going to be able to balance their minerals. The salinity, the salting of soil that is such a huge problem all over the world, they're going to start mitigating that and reversing that. They're going to be able to have the biology, because organic matter contains the biology to begin with. And they're going to be able to sequester the carbon. And so all the benefits will all occur. And then the nutrient density of plants goes hand in hand with all of those benefits. So it's going to cause the biology to return, and the biology to probably come to places that hasn't been there in hundreds, maybe even thousands of years. Because I mean, like, that's, the birthplace of agriculture is a desert now, despite it being fertile once upon a time, which means it can be fertile again. So this is the way that we expose and pull this stuff out of the dark and allow for best practices to rise to the top, for us to get the skills that we need to communicate with each other as peers in a process of community-based solutions that allows for, and everyone's part of the community rating system. So you're rating the entries that you're reading and checking out, and that makes it so that we really have that ability to self-regulate. So I'm really excited about it. People are going to be able to learn and grow. They're going to be able to be recognized. They're going to be able to select and find those farmers in their area that are doing the right thing. They're like, wow, what are they doing? They're having a farm tour next week. Let's go.
And imagine the farmer who looks at the database to see who's at the top. What are they doing? How can we do that?
Right. And within 5 to 10 years, if we get everyone involved and we get these things to happen, it is going to change all of soil science, all of farming, all of food. And then, because food really is medicine when it's grown properly, it's going to change all health. And from there, I mean, once it's a proven concept, it's like, oh, it's the right microbes, it's the right organic matter. It's like, then we know how to fix all the bioregions. Then we know how to fix the oceans, because oceans have their own soil and their own redox and pH, right? It's all just that simple. And so we just need to balance these things, bring back the biology, bring back the buffering capacities of the soil, of the ocean, and so much of it's fungal.
Which I love to hear. But just thinking of that bioregions, imagining mapping public health data from that bioregion, you can see over time as these soil compositions are changing, what's going on. Yeah, that's like the ultimate goal. And it sounds like a lot of where it started for you was very much grounded in health and where do the answers, and you got to go all the way down under the floor, into the mycelium underground. And I was curious, how all of this work, and not to make it too broad a question, but how all of this work really has empowered you, in all of those human roles as a father, as a partner, as a teacher, this work in understanding something as fundamental and as elemental as soil and the components of soil and diving into these microscopic worlds. How does that then, that's the classic kind of alchemical proverb, is by doing that work, your insides are changed similarly to how you're doing your outside activities. So how has that changed you and shaped those relationships and who you are as a person?
The world that I live in, because of this information, is completely different from the world I grew up in, from the 80s, where I was powerless and had no role in it. And I was just a victim of circumstance and people in power around me. Hmm. The information that I've gained from all this study is that we are actively affecting everything around us constantly. And that we have an opportunity to make a massive impact. And that impact goes in as it goes out. And as we change the world around us, we change the world within us. And that's why meditation also works. You change the world within, you change the world without. But also, there's nothing compared to action. Nothing compared to right action. Doing the rightest action that you can take in a moment. Something so powerful about that. And for being a dad, I am a family guy. I think about the fact that I'm raising boys all the time and how they're looking at me and thinking about how they're going to be as a dad, how they're going to be as a husband, how they're going to be as a man. And so I think about that all the time. And when they were younger, I used to be so scared about the world that they were inheriting. And yeah, there's adversarial craziness going on in the world, man. Oof, it's rough. It's rough. But at the same time, it's always been rough. And at the same time, we never knew what we know now. And my boys see that, because of the way I understand the world, I approach the world with total faith and confidence. And my son, James, my older boy, he literally manifests things all the time. He came home with 6 foot, maybe 5.5 foot tall speakers 2 days ago. And I'm like, your luck is back in. He was like, yeah. It's amazing when these things become aligned in a person's life. And so I'm really, really keen about keeping myself like a temple, keeping my mind like a temple, trying to keep my life as pristine as possible, and just being grateful. So yeah, this work has completely changed the way I view the world. The world is so beautifully complex and mysterious and interesting. And we're about to unlock it at such a high level. Just the fact that we measure soil now, after Olivier Housson, and now with my book, Regenerative Soil, Redox, and we're measuring the millivolts of soil, which is, oxidation is the loss of energy, reduction is the gain of energy, redox. And when we are taking antioxidants, these are highly reduced foods, which means that they're high in energy. And it's like, okay, so everything is energy. And then it's like, everything's crystals too. Microbes can turn into crystals and actually reform back into microbes, they've discovered recently. So it's like, yeah. And all the plants look like they're made of glass. They look crystalline under proper light, under real light.
And fungi have crystals.
They form crystals because they're digesting everything externally, and they really do look like crystals too. They're absolutely incredible. So it's an incredible world that we live in, and we're just scratching the surface of what's true. But we can give each other the ability to understand, interpret, and discuss these things and navigate this space. And that's really what needs to happen, rather than like what you can find some places where they're like, no, this is the way it is, and this is this, and this is that, and we know this, and it's not like that. It's the Wild West, and it's beautiful. And there's so much to learn and grow in this space, but we have to do it in a way that's transparent. And a lot of people don't like that. But too bad, because the reality is this is the only way that we're gonna really fix soil fast. This is the only way that we're gonna get the best food fastest. And this is how we honor all those people that are trying so hard, doing the right thing in obscurity in their backyard with this amazing fungal compost or Korean natural farming. It's like, this is the opportunity for all of them to be like, oh yeah, check this out. It's like, do it. Let's have it happen. Let's get everyone excited, because this top-down narrative that exists out there that like, oh no, just trust us and we'll save the world and take your rights at the same time. That's like,
How's that been working?
yeah, yeah. I'm actually really worried about those Dutch farmers. They're shooting at Dutch farmers right now. There were shots fired at their protest by the cops.
Yeah.
And so we're the heroes of the story. We are the only ones that can actually fix this. We're the only ones that actually can buy the right things so that we shift the market. Like,
And that's why I think it's so important to support this Kickstarter project, but really this concept, because you said transparency is kind of sometimes seen as a threat. Well, let's pour all of our collective energy and intention into something that's doing just that and really force this out into the open, elevate this kind of project for all the beautiful reasons that you elucidated and the empowerment. And, a lot of times we talk about, manifesting and things being magical. It's like, well, if you're going to quote unquote work magic, you need to know what all the ingredients are. And this is part of that, is getting the skills and understanding how things work. And the way you described the world, I had a feeling this just informs an entirely different worldview. There are people like yourself, people like Will, people that go deep into the biological world that you can tell when they come back, they're seeing a different planet than I am. They're seeing a different reality than I am. So it's also a clarion call, not only to get involved and support the Kickstarter, but also, and partially saying this to myself, so I hear this, but to get involved in these disciplines. No better time to learn than now, and start to learn this process, get a copy of the book, chip away, because it sounds like, this is something that should be in the skill set of whoever you are. You should have a working familiarity with this stuff.
yeah, and to go really tangential, folks are like, half these people that like are working in microscopes are actually nurses, and they're testing their blood, and they're figuring out that like certain foods allow their blood to flow freely, like their cells to like each have a charge fully around themselves and to bounce like balls, and then other foods cause them to clump into these structures that clog up our veins.
Oh, man.
Yeah. And so like people are like using their microscopes in a variety of ways that I'm not teaching and I haven't done yet, but they're showing me and I'm like, wait a second, this is crazy. And they're drinking fourth phase water.
What is fourth phase water? I've heard
Gel
this
state
term now
water.
like twice.
So gel state water is fourth phase water. So if you take like sodium alginate powder and put it in a blender, it makes a gel. People are using that for beauty, because gel state water actually makes you much more hydrated skin wise. So it's less, you wrinkle less. Yeah, no, this is real. But it also literally makes it so that it changes the charge on the outside of your cells, your blood cells. And this is also why Zach Bush cells restore that gut biome solution. It's actually, if you read the site on it, it's terrahydrite, earth water. And it's basically a very, very refined compost tea that's just minerals. So they're like prebiotics in a way. So they feed the biology in our gut. And that changes the charge on the outside of our cells. That's how he advertises it. And that's literally what fourth phase water does too. They're doing that. And so I think that we're all actually going to start using microscopes and community microscopes like this. Like, this is an expensive microscope. I'm showing ways that we can use cheaper microscopes to do exactly this. But nicer microscopes, community labs should all have them. We should train our kids on how to use them. My 11-year-old boy can use this, and I trust him with it. This new, and all of my students get a 40% discount from this company, so that's I got the same. But it's $6,000 new, and my 11-year-old use it. Why? Because he can. And it's like, that's the thing. It's like, you can too. And it's unbelievably fascinating. And just, you can see health. You can see how inoculated your plant is. Like, instantly. You can take a root without any cover slip and just stick it under there, flip on that blue light, and instantly see it. That's what this is. And there's no cover slip. There's no preparation. I just put it on there and I can see if it's inoculated or not. And I already know this isn't inoculated because it's amaranth, but I was...
One of the few that isn't.
Yeah, non-mycorrhizal. I actually did an experiment where I put a mycorrhizal inoculant on it, and it tried to persist and consume the exudates on the outside, produced a few like crystals, and then senesced. So it was like fascinating to see it try and then just not get in.
I'm just thinking this makes the relationship with science and so many of these concepts that much more tangible, where you're looking at it. Again, it's not numbers. It's not something where solvents were applied and you got a spectrometry reading or something like that. It's like you are seeing it. You are in that world, getting the definitive answers. So, man, the new tool of our age, that's the real new wave, is getting in a microscope. And I love that you referenced Zach Bush too. He's like one of my favorite people. So yeah, everybody needs to get microscopes, get into soil, support Matt's work, follow Zach Bush. And just one more time, plug, where can people find the Kickstarter? What do we type in? How do we get there?
Okay, if you go to Kickstarter.com, you can type in my name, Matt Powers, or Regenerative Soil, and it'll just pull up the rest. We're one of their featured projects we love. So Kickstarter has been promoting it, which is epic. And yeah, we got 10 days left. Maybe when this airs, even fewer days. But check it out. It closes 8 a.m. July 17th, Central Time. So
call to action, get on it, guys. Well, and I normally have a couple questions I ask all my guests, kind of boilerplate. I'll boil it down to just one. From the man who swims in soil and has these superpowers, sees the world of crystals, and what is a fungus that you love and why? And that doesn't have to be a big showy mushroom, doesn't have to be something you eat, but a mushroom you love, or a fungus you love, and why?
Arbuscular mycorrhizal fungi. I love it because it partners with over 90% of all plants. It sticks around for only a few days or up to a couple weeks, and then it leaves, and it collects the nuclei of other fungi and other species. And when it creates spores, it can spin out whatever it wants in relationship to the current environment and the level of secession. So it can cue secession, and it can react to the environment. And it's everywhere. And we don't know its ancestor. It's such a mystery. It's so powerful. It's all in service. And it's flying in the air. It's amazing.
Leave it to you to make something AM fungi that so many people into mycology just take for at its face value, and to deepen our appreciation and show us the magic behind that. Well, Matt, thank you for taking us into the world of microscopy, crystals, fungi, plants, nematodes. It's been a fascinating conversation, super inspiring. I'm really excited to follow your work. Thank you for coming on the show.
Thank you so much for having me.